DNA. Definition. DNA is an informational molecular encoding the genetic instruction used in development and functioning of non-living organisms and many viruses. Along with RNA and proteins, DNA is one of the three major macromolecules that are essential for a known form of life. Structure. Genetic information is encoded as a sequence of nucleotides recorded using the letters G, A, T, and C. Most DNA molecules are double-stranded helix consisting of two long polymers of simple units called nucleotides. Molecules with backbones made of alternating sugars and phosphatase groups with the nucleic bases attached to the sugars. DNA is well suited for biological information storage since the DNA backbone is resistant to cleavage and the double-stranded structure provides the molecule with a built-in duplicate of the encoded information.
DNA contains two strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes three parts: a phosphate group, a sugar molecule, and one of four bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
DNA replication. Replication is the process where DNA makes a copy of itself. Why does DNA need to copy? Simple. Cells divide for an organism to work and reproduce. Every new cell needs a copy of the DNA, of instructions to know how to be a cell. DNA replicates right before the cell divides. DNA replication is a semi-conservative technique that it makes a copy one-half of the old strand. If overtaken the new strand, this helps reduce the number of copy errors.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Genetic engineering methods have been developed to purify DNA from organisms such as phenylchloroform, extraction, and to manipulate it in the laboratory, such as restriction dejects and the polymerase chain reaction. Modern biology and biochemistry make intensive use of these techniques in recombinant DNA technology. Recombinant DNA is a man-made DNA sequence that has been assembled from other DNA sequences. They can be transformed into organisms in the form of plasmid or in the proprietary format by using of viral vector. The genetically modified organism products can be used to produce Products such as recombinant proteins used in medical research or behavioral in agriculture. DNA nanotechnology use the unique molecular recognition properties of DNA and other nucleic acids to create self-assembling branch DNA complex with useful properties. DNA is the use of a structural material rather than of a carrier of biological information. This has led to the creation of two-dimensional periodic lists, as well as three-dimensional structures in the choice of polyhedra, nanomechanical devices or algorithmic cell assembly have also been demonstrated, and this DNA structure has been used to template the arrangement of other molecules such as gold nanoparticles and extractable proteins. Advantage: increased likelihood of finding asylum on criminal offenses, especially violent and sexual assault. Trying to solve or unsolved crimes. Biology further identified with very high probability leads to solution in wide range of family problems. Identification of victims in mass terror attacks, cracks, accidents, etc. Familiar searching and analysis increases the probability of criminal detection. Disadvantage: six as an infringement of civil liberties, especially when DNA profiles are stored from people who have nothing to do with a particular criminal offense. Concerns that the finding to authorize amounts of DNA can falsely implicate a person in a criminal investigation. Concerns that the complexity of testing and data stored means error could occur. DNA profiles could be related to certain illness and character traits, and this information will be used by insurance companies and other organizations to the disadvantage of the individual.